morning, everybody. I think we're going to go ahead and get started. It is noon. So welcome to the Restoring the West conference. I hope you all can hear me. My name is Gabrielle Harden. I am the Forestry Extension Educator at Utah State University. I'm going to moderate for us today. If you have any questions for the speakers during our presentations, please add those questions in the Q&A box. Here to kick us off today is Karen Mock. Karen is the department head of Wildland Resources at Utah State University. She earned her PhD in biology from Northern Arizona University. Her research emphasizes in genetics and conservation and management, and much of her research at USU has been in Aspen. So Karen, I'm going to go ahead and hand it over to you.
Hi there, can you hear me all right? Good. Hi everybody, and welcome to the COVID version of Restoring the West. I've always really enjoyed these conferences. And we'll miss seeing people in person this year. This conference to me, as a department head especially, is the embodiment of what a land-grant university should be doing, to bringing science and land managers together so that our research has a positive impact on the ground. And I think both research and management are greatly improved by this kind of crossover. So, and a special thank you to Darren for pulling this together despite the odds and the ridiculously uncertain times. I look forward to hearing from all of these speakers, and I hope next year we'll be able to have something a little bit more in person. So with that, I'll turn it over to Darren.
Thanks, Karen. I think, Gabrielle, you need to turn on my video, please. Thank you very much, Karen. Appreciate it. And welcome everybody to the 2020 version of Restoring the West. We're calling it Restoring at Lunch because we're mixing it together with our traditional Learn at Lunch webinar that we started in 2009, our monthly webinar series. And want to welcome everybody and really appreciate you tuning in to this virtual conference and working along with us today. If you were in Logan, you would be here for a beautiful day. It's a little bit smoky, but the maples are changing. They're not quite as orange as they are in some years, but it seems like in the deep canyons, they're redder than they even normally are. I wanted to start off by thanking our committee, the Restoring the West Planning Committee, and that includes Mike Kuntz and Gabrielle Harden, who you've been introduced to. It also includes Paul Rogers, Mark Brunson, Larissa Yoakum, and Justin DeRose. And we couldn't do it without everybody's help, that's for sure. Also want to thank our sponsors today. And those include USU Forestry Extension and USU Department of Wildland Resources, the Quinney College of Natural Resources, and USU Cooperative Extension, the USU Ecology Center, the Southern Rockies Fire Science Network, and the Great Basin Fire Science Exchange, the Western Aspen Alliance, the Utah Biomass Resources Group, and the Utah Prescribed Fire Council. Want to thank all of our sponsors. And we'll move on with our first presentation. We're excited to be focused on soils and on aspen over the next 2 days. And these are super important topics and often come together on certain projects, some that I'm working on right now. One in particular is called the 106 reforestation method. And here in a month or so, you can tune back in and we'll be hosting a virtual version of my annual timber harvest tour and wildland prescribed fire tour as we examine this new roller felling method of disturbance that's being developed here in Utah. But on to today's first speaker. We're really pleased to have Nichole Barger speaking with us today from the University of Colorado. Nichole is a professor of ecology and evolutionary biology. And she's a dry land system ecologist that focuses on resilience of ecosystems from in regards to anthropogenic and natural disturbances. Gabrielle, if you can turn it over to Nichole, thank you very much.
Hello, everyone. Thank you, Darren, for the great introduction, and thanks to the whole committee today. I hope everyone out there is doing well, and this is our new normal. So what I'd like to do is, it looks like I'm not quite able to share my screen yet, so I need to have that enabled before I can start. If someone can do that on your end.
You should be good to go. I'm sorry about that.
Okay, thank you. All right, so like Darren said, I'm Nichole Barger. I'm from the University of Colorado, and today what I'm going to be talking about is restoring Western soils and really focusing on some of the successes and challenges in biological soil crust restoration. I have been working on this with a team of scientists since 2012, and this is a really exciting new area of research. And we've been thinking a lot about how we, you know, from when we started doing this research, we're trying to understand how biological soil crusts recover, what we can do to enhance their recovery, but also really be thinking about how land managers can really use this information. So I'll be interested in the Q&A session to get your feedback on, you know, if any of you have tried any of these techniques or are thinking about it or interested. So first I want to acknowledge that all of this work today took a huge team of scientists from not only from the University of Colorado and my research lab, but from the US Geological Survey, Northern Arizona University, and Arizona State University. So as many of you know, we have a lot of soil conservation concerns in the Western US, especially in these arid land environments where we have a lot of human activities, such as oil and gas development, livestock grazing, recreational activities, energy development. And what we know is that a lot of this can have pretty significant impacts on nutrient cycling, hydrologic function of these ecosystems, soil erosion. And what we also know is that biological soil crusts play a really important role in the functioning of dryland soils, especially across the Western US, and in particular, the Great Basin and the Colorado Plateau. So biological soil crusts, for those of you who haven't thought a lot about these communities as I have, are just communities of cyanobacteria, lichens, fungi, and mosses, and other bryophytes that colonize those top few millimeters of soil and are important, especially for soil stability in these systems and reducing soil erosion. So an important component of soil recovery after disturbance in these environments is really contingent and on the recovery of the biological soil crust community. As many of you also know, biocrust recovery may also be fairly slow after disturbance. So what we've really been thinking about is how do we give these really important biocrust communities a shot in the arm in terms of their recovery to promote a faster recovery time. So the framework that we've really thought about in terms of how we link restoration of soils is we can think about this as a biocrust adaptive restoration framework. And if you want to play with acronyms, that would be, we can call it BARF. Probably not the best acronym, but it sticks in people's minds. So what we can think about is that a lot of times if we have a stressor in the system, we can just simply remove that stressor and hope for some recovery. But what we know is that that is often slow. And this is likely, as we move up this, into more active restoration, it's likely due to the lack of, in our case, the lack of biocrust propagules in the system. So what we can think about is, well, how do we get propagules or seeds into the system for biocrust recovery? But we also know that in highly disturbed soils is that we may need to engineer the site a little bit more for that inoculation to take. So site engineering, such as adding nutrients or stabilizing soils, also can be a really important component. So this is the sort of framework that we took in this project that I'll be describing. So we took the staged approach to our objective to facilitate the recovery of degraded arid and semi-arid land soils by restoring biocrusts. by first developing BioCrust inoculum in these nurseries, while we also just at the same time tried in the field a bunch of different candidate approaches to restoring BioCrust. And then these two approaches then came together into these larger experiments to evaluate soil responses to BioCrust restoration. So I'm going to step through each one of these, and the project that I'm going to describe today took place in cold desert sites in the Great Basin at the Utah Test and Training Range, and also in hot desert sites in the Jornada Experimental Range. Given where we are, what I'll be doing is presenting results just from our cold desert today. So we took two paths to restoration of BioCrust, and each one started with collecting native small amounts of field collected BioCrust samples. And one path was to just take these samples into the greenhouse and then increase their biomass in these greenhouse grown BioCrusts. Then that can then be deployed into our field experiments. Another path and a more intensive path was taking field collected BioCrust, culturing some of those organisms, and then scaling that biomass to field experiments. So as you can imagine, there are trade-offs in developing these different types of BioCrust inoculum. So going from left to right here of inoculum types, there's where you can just collect BioCrust in the field and use it in your restoration of a degraded soil. But what we know is that if we just take BioCrust from one location and try to restore another, we have a high risk of over-harvesting. Whereas if we take small amounts and grow them out in the greenhouse, we're able to increase that biomass without a lot of risk of over-harvesting, and even more so when we take very, very small cultures and then increase that biomass. However, the trade-off is that as you move from left to right, from field collected to local biomass to mixed isolates, you have organisms that are less adapted to the local environment. And also the time and resource investment increases as you move from left to right. So what I'd first like to describe is the cultivation of mixed isolates, which was work led by ASU by Ana Geraldo Silva. And these are some of the species that we were cultivating in these isolating and culturing and then cultivating. And this was a pretty time-intensive process, which could probably be increased in efficiency over time, but this was our first try at increasing the biomass. And it took approximately 6 months to go to a very small amount of cultured isolates to about 20 liters 20 liters of biomass that we could use then to deploy into our field experiments. So this 20 liters of cyanobacteria that we cultured was then mixed with native soil, and some of them had to be grown on filter paper. So as you can see in this right photo, you'll see that there's some filter paper on top, which some of these cyanobacteria were only able to grow on those filter paper. So then when we went into the greenhouse from the second pathway of developing BioCrust inoculum, so we had these field collected BioCrust samples, we grew them out in these small containers that were frequently watered in these greenhouse environments. And after about 3 to 4 months, that BioCrust was then scraped, dried, and then deployed into the field. So what we found in the greenhouse experiments, what was really interesting is that we could really increase the biomass quite quickly. And so what I'm showing here with this red line, this is 2 different soil types from our Great Basin sites, a clay loam and a sandy loam, is that those field collected samples came in with about 1 microgram per gram soil of chlorophyll A. Chlorophyll A is an important indicator that I'll keep showing because it shows the photosynthetic capacity of our soil and of our cyanobacteria. So we were able to increase the biomass by nearly 50 to 60 fold by growing them up in the greenhouse. So we had great success in doing this. So what we were also interested in is that as we were growing these small field collected samples in the greenhouse, whether or not we were getting a lot of weedy microbial species coming in. And so what we're showing here is looking at what was the microbial composition from our field collected samples here on the left. And so you can see that there's, in the red, there's a lot of cyanobacteria. And then on the right, on the left graph, but the right bar, what you can see is that the cyanobacteria increased in relative abundance in our greenhouse samples or in our greenhouse grown samples. And so what we generally saw, it's the same pattern in the left graph, is that the right graph is that the community composition was generally stable. We did have some weedy species coming in from the greenhouse, but we were pretty happy with being able to maintain that some of those native species that came in from the field collected samples. So the other thing we did at the same time that we were developing this BioCrust inoculum is that we were identifying the best candidate BioCrust restoration strategies. So we had a lot of different approaches of adding nutrients, stabilizing soils, shading the soils, and just to implement a lot of different strategies to see what would work. So I guess this is, fortunately, we had some really positive results, but unfortunately, from a management perspective, this was an interesting result. So our most promising approach to restoration of BioCrust in this full suite of things that we tried is that shading and inoculating with field collected BioCrust, and this was led by Taylor Schock, one of my graduate students, was shading and inoculating. So what I'm showing here in this graph, again, this is chlorophyll A, and on the left-hand side, this is a fully scraped soil. There is no BioCrust there. And then the right-hand side, we can think of that as our target, that this is about the amount of chlorophyll A that we're looking for, and that's in these intact BioCrusts nearby. So what we saw over 3 years is that the NR is that nothing has been done. There's no shading or no inoculum. But as you can see, the shading plus inoculum has had the greatest success. So the unfortunate part of these data is that going out and asking managers to shade all of their soil for BioCrust recovery is not going to be the most tenable solution. But for smaller scale projects, this could be quite a successful approach for smaller scale projects, but likely not a good approach for landscape level projects. So what we did do in this final large multifactorial experiment where we looked at all the information coming from those best candidate approaches is that what we realized is that we really needed to stabilize our soils first. And this work was led by Akasha Feist, who was a postdoc in my lab at that time. And so we had 2 different ways that we stabilize soils. One was with these 1 meter squared straw checkerboards, and you can almost think of these as small fences to keep those soils from moving in the high winds and with high water erosion. And that is being shown on the left. And then the landscape scale of the straw checkerboards in China is shown on right below that. The other thing that we did was we tried these soil stabilization products that are commercially available, these polyacrylamides, to first stabilize soils and then inoculate. So the full experimental design was that we had intact controls as our target. We had, on the left, we just then disturbed all of our soils. We also had a control that was just disturbed, uninoculated. Then we added no inoculation with our polyacrylamides, but only stabilized with our polyacrylamides in straw. And then we had our field collected local biomass grown in the greenhouse, mixed isolates grown in the lab with both stabilization approaches. So here's those data. Again, we're looking at chlorophyll A, and on the left, you can think about this as our target chlorophyll A for intact BioCrust over those 3 years. So looking at this, you have all of our treatments across here. You look at this and go, huh, wow, there doesn't look like to be much difference. And so this was a little deflating for us at first, trying to understand, how the system is working when we've inoculated with these different, we've stabilized these soils, we've inoculated with these different types of inoculum that have had different methods. But if we go in a little bit, what we can see is that we are starting to see after 3 years some differences in chlorophyll A. And so maybe in some ways this isn't too surprising, is that our field collected is showing some, starting to pull apart and showing some difference. And this is likely due to the fact that these field collected just immediately came from the field. They haven't been in the greenhouse, they haven't been in the lab, and they're likely better adapted to really taking a hold in these degraded soils. So the other thing, again, that we were interested in is this is just another measure of cover of the full biocrust community and not just the cyanos. And so what we see here on both of these graphs in 2015 and 2017, so 1 and 3 years after our experiments, is that we have this full community of nitrogen fixers in the system. In 2015, 1 year after inoculation, we had a lot of bare soil still, but interestingly, we started seeing not only the light cyanobacteria, the microcoleus, but we're starting to see in some of these, with the brown bars on some of the treatments, some of the nitrogen fixing cyanobacteria coming in. So the other thing I want to show as the last data slide is the soil aggregate stability, and really thinking about how we start, if you are working on a degraded soil in these environments, how we start really stabilizing these soils. And those red dots, what I'm showing is the polyacrylamides, and thinking about how we can start using some of these commercially available products for soil stabilization. So the highest level of soil aggregate stability class is a 6. And as you can see, relative to the other treatments, these red dots or these polyacrylamides really maintained their stability early in our experiments. So some key messages from my talk is that we have developed 6 pretty simple, quick, and effective methods now exist to grow biocrust inoculum. The mixed isolate isn't so quick, and you can think about that more as a method that could be developed commercially. But there are, in terms of the skill that would be needed to take a field collected biocrust and grow it up in a nursery in a greenhouse, it's pretty simple, and you can do that pretty quickly. And there are more time-consuming lab culture methods that we are still working on, and hopefully over the next 10 years, these will be more commercially available, like some of the mycorrhizal inoculum that we see. If you were to do some smaller scale projects and you want to get biocrust recovery going very quickly, shading is a pretty effective method, plus inoculation. But at this point, there's been a huge boom right now in trying to understand biocrust restoration and recovery. And so we still are experiencing challenges, but my hope is that over the next couple of years, as this research becomes of interest to a lot of different researchers, that we'll see more and more success. So with that, I'd like to, this work was supported by the Strategic Environmental Research and Development Program from the Department of Defense, and was supported by a huge number of undergraduates. So with that, I hope I have time for a few questions.
Yes, you do have some time for a couple of questions, and then any ones that we don't get to, we can address in the panel Q&A at the end. So Eric McCauley would like to know if these methods have been used, oh, it went away, have been used in stream bank restoration to encourage riparian areas to reestablish.
That's a really great question. And Sasha Reed from the USGS and her previous postdoc, Colin Tucker, they have done some restoration in some greasewood areas down south near Canyonlands National Park, Needles District, very close to a riparian zone, and they'll just be getting some information, I hope, over the next year on that. But, you know, in a full, I also know there's another Lake Powell study, and I can share this as as I am here later today. I can share some of these resources. But there is also a lake restoration study. I hope that answers your question.
Okay, so it looks like we're approaching that time where I've got to move on to Erin, but I'm going to save, there's quite a few more questions for you, so I'm going to save those, bring them back out near the end. So thank you so much for taking the time to speak with us today. We really enjoyed your presentation. Erin Hanan is our second speaker today. Erin is an assistant professor of fire and ecosystem ecology at the University of Nevada, Reno. She earned her PhD in ecology, evolution, and marine biology at the University of California, Santa Barbara. She studies interactions among plant, soil, and hydrologic processes in the terrestrial systems. Her topic today is wildfire effects on nitrogen cycling and export in dryland watersheds. So Erin, over to you.
Thank you. Thanks for the introduction, and also thanks to all the organizers and to everybody who's here. I am just trying to share my screen. Let's see. You'd think I would have this down by now.
That's just fine. Technology is always
Sorry, I know, I've
day.
lost my window. Oh, here we go. Sorry about that, everybody. Give me 1 second. Okay, how's that?
Looks good.
Okay, thanks. So today I'm going to be talking about some of my research on nitrogen cycling and export in fire-prone watersheds. So I basically research in my lab has a couple different focuses. For one thing, I'm interested in things like how climate change and management activities influence wildfire regimes across the Western US. But then I'm also interested in how changing fire regimes affect ecosystem and watershed processes. And that includes things like carbon balance, nitrogen cycling, and stream flow. And I'm especially interested in how these processes interact across scales, ranging from ecosystems to entire watersheds. So some of the Western systems, and sorry if there are cats zooming around in the background, some of the Western systems that I work in include coastal California chaparral, inland Northwest mixed pine forests, so including the Northern Rockies, and then Sierra Nevada and the Great Basin. And a lot of what I'm talking today focuses on nitrogen cycling studies that I've done in California chaparral. So we know that globally, many ecosystems are dominated by fire. And this is illustrated really well in this figure where the color intensity is related to fire activity. And so it's important to understand how fire affects ecosystem and watershed processes. So fire is a major restructuring force in dryland systems, and it can cause massive redistribution of materials both within and among landscapes. So for example, during fire, large quantities of nutrients, especially nitrogen, are released from the vegetation, from litter, and from surface soils. And a lot of this nitrogen is volatilized and transported downslope. And so nutrients can be carried from, pretty far inland to way offshore. And that is shown in, this is an image from NASA, which was taken during the 2003 firestorm. And so you can just see that a lot of materials, including nitrogen, are taken away during fire. So in addition, water can be a really powerful vehicle of post-fire export. And so this picture was taken after the debris flow that destroyed Montecito and San Ysidro Creeks after the 2017-2018 Thomas fire. And so this happened 3 weeks after the fire, and a single rainstorm that lasted only about 20 minutes basically just completely devastated the downslope community. And I've heard Ed Keller at UCSB describe this as like ping pong balls floating in a bathtub. The mud and debris becomes so dense that these like many ton boulders just bobble through. So it's very intense. But even in the absence of this sort of intense debris flow, erosion and leaching can really threaten water quality and aquatic systems. So we know that nitrogen is a critical nutrient for recovering plants after fire, but it acts as a pollutant in streams and reservoirs or the nearshore marine environment or lakes. And so seasonal pulses of nutrient export can sometimes occur for multiple years after fire. And the length of time and the amount of nutrients and nitrogen that's exported is a function of how severely fire altered ecosystem and watershed function, and then how quickly ecosystems recover and how quickly vegetation is able to begin to immobilize that and that is released during fire. So a key question motivating my research is, how does fire affect ecosystem and watershed function? And so a key ecosystem function is nitrogen cycling. And so again, as I just mentioned, nitrogen is a critical nutrient for recovering plants, but it can act as a pollutant in streams and reservoirs. And so as an example, this photo shows the Whittier fire that burned through the Santa Barbara backcountry above Lake Cachuma in 2017, I think it was. And so Lake Cachuma is the main reservoir for Santa Barbara County. And so this fire really, as you can see it burning way down close to the reservoir, it created a lot of concern that runoff from the burn slopes could devastate local water quality, drinking water supply. And these concerns are growing with climate change, since fire and precipitation regimes are already becoming increasingly variable and more severe. So this kind of leads to a couple important questions. One is, what mechanisms control nitrogen cycling and retention as ecosystems recover from fire? And two, how does climate affect the magnitude of post-fire nitrogen export? So I will talk about some of the research I did answering these questions in coastal chaparral watersheds. This photo is at the top of Rattlesnake Canyon, which is just inland from Santa Barbara in the Santa Ynez Mountains. So coastal chaparral ecosystems are really tightly connected with streams and with the nearshore marine environment. And so these ecosystems are characterized by dense stands of evergreen shrubs. These shrubs grow rapidly in winter and then become highly flammable during the hot, dry summers that are typical of California's Mediterranean climate. So as I showed in the introduction, a lot of nitrogen is volatilized and lost during fire in chaparral, but at the same time, fire deposits nitrogen on soil surfaces with ash. And so that's usually in the form of ammonium and or readily decomposable organic N. And so the issue with this is that winter rain can erode soil and leach nutrients, especially the nitrogen, into streams and reservoirs or the nearshore environments. And this is especially true if a slope has been denuded by fire. And so again, this can be problematic since excessive N acts as a pollutant in aquatic systems. But at the same time, nutrients promote the germination and growth of ephemeral herbs. So this photo is, this is mostly Calistegia macrostegia, which is a native morning glory species that comes in really rapidly after fire. And so these ephemeral herbaceous species that come in quickly and are fast growing, they play a really important role in taking up and rotating, sorry, my cats are going crazy. They play an important role in taking up and immobilizing nitrogen as the shrubs recover more slowly. So recovering shrubs tend to replace the ephemeral herbaceous species within the first several years of recovery. And within about 10 years, you tend to have a closed canopy. So chaparral shrubs recover fairly quickly compared to a forested system. And so the extent that nitrogen is transported from the burned slopes to streams over the course of this recovery process is a function of how quickly soil microbes metabolize nitrogen into forms that readily dissolve or leach, and how fast recovering plants take up this mineral N. And each of these processes can vary over the course of recovery, and the balance between them can also vary a lot in response to climate change and drought. So one very robust pattern that I found and many people have found is that fire stimulates net nitrification, which is the microbial oxidation of ammonium to nitrate. And nitrate rarely accumulates in mature chaparral that hasn't burned in a long time. And rapid nitrification can be problematic because, one, it can intensify greenhouse gas emissions, and it can increase nitrogen leaching since nitrate is particularly mobile in the soil. So I did a laboratory incubation experiment, and what I found is that nitrification is enhanced by interactions between ammonium addition, which occurs with ash deposition, and increases in soil pH. And this happens because, one, fires destroy organic acids in the soil, and two, fire deposits these base-forming cations on the soil surface with ash. So pH tends to increase after fire. And I found that these two drivers can trade off or amplify one another and create really optimal conditions for nitrifiers to thrive. And so as a note, these are soils where the native nitrifier populations are low. And so once you build up a population, that population can persist and continue to nitrify even after the soil pH and ammonium have returned to their pre-fire state. So you can have these lasting effects. So although these studies provide quantitative measurements of the mechanisms that are interacting to increase nitrification, what I couldn't account for in field and laboratory studies is the relationship with some of the other things that change after fire. So for one, there's a really large potential for recovering plants to take up mobilized nitrate, as I showed you with that picture of the Calistegia. And two, climate conditions that surround a given fire can influence the rates of nitrogen mobilization and nitrogen uptake. So an important question is whether these are big or small numbers compared to the effects of ammonium and pH in increasing nitrification. But to answer that, we really need other tools. So we need models. So I work with a model called RHESIS, the Regional Hydroecologic Simulation System. This is a process-based land surface model that aggregates data from several spatial, geospatial layers to represent the distribution of topography, vegetation, soils, and hydrologic characteristics in a watershed. And so then we feed climate inputs or meteorological inputs into these process-based submodels that calculate carbon, nitrogen, and water fluxes across this complex terrain at multiple scales. And so we're simulating processes like stream flow, net primary productivity, nitrogen cycling, nitrogen export. And one of the strengths of this particular modeling framework is that it tightly couples both hydrologic and biogeochemical processes over space and time. And so that makes it really ideal for representing carbon, nitrogen, and water fluxes in drylands or mountainous watersheds, because these fluxes in plant growth are water limited. So we need models to convert our assumptions about a system into projections, but we know that models are really just elaborate hypotheses. So why do we believe a model like Rhesus? This model has stood up to a lot of challenges. So this map, and this map is outdated, but here's a map that shows many of the places that Rhesus has been run and evaluated and refined using real observational data. And so in Chaparral, Rhesus has been extensively tested against things like stream flow, nitrate export, and remote sensing of vegetation. Other systems, it's been tested against things like tree ring data, data from flux towers, and measurements of photosynthesis. So although the model is very well developed for watershed biogeochemical processes, it did need some modifications at the time to effectively capture fire. And so I did several things to represent fire in the landscape, just including removing above ground vegetation and litter carbon and nitrogen stores, added ammonium to the soil surface, reduced hydraulic conductivity, increased pH, and so on. And so my big question is, how do climate conditions before and after fire influence nitrogen cycling and nitrogen retention? And so I set up a modeling experiment where I replaced the precipitation data for 4 years before and 4 years after fire using the driest and wettest years on record. So to simulate, like, if you have a wet year versus a drought year and how that influences post-fire processes. And so in this graph, I'm going to show nitrate export on the Y for 2 years before and 2 years after a fire event. And I'm going to start by showing simulations where fire was followed by a relatively wet year. So in one, it was dry before, in one, it was wet before. But the key thing here is that it was wet after fire. And in both cases, most nitrogen export occurred during the first wet season after fire in a pulse. And the pulse was slightly larger when fire was also preceded by wet conditions. So an interesting and sort of surprising thing was what happens when fire is followed by drought. So the red and the purple lines are showing basically when fire is followed by drought. And so for one thing, there's a few things going on here. For one, you can see that the pulse of N during the first post-fire year was delayed here due to a later onset of the winter rainy season. And you can also see that in both cases, there was a second pulse of nitrogen to the stream during the second year of recovery, which led to more total nitrogen export. And this occurred because drought suppressed plant recovery and nitrogen assimilation relative to rates of mineralization and nitrification. And so overall, what I found is that in this particular chaparral system, nitrogen export was highest when fire was followed by drought. And this might seem somewhat surprising since globally nitrogen export tends to increase with increasing precipitation. But at this scale, because soil microbes are less sensitive than plants are to drought, dry conditions following fire allowed the mineral nitrogen to accumulate over the hot, dry summer and then be flushed from the soils when wet up occurred. So one way that climate really influences nitrogen cycling is by regulating how plants recover following fire. So just to describe some ongoing research in my group. So we know that nitrogen export to streams has key implications for water quality. It can also be an indicator of ecosystem watershed health. So historically, nitrogen export has been used to identify when a watershed is nitrogen saturated, or the point where nitrogen inputs from things like fossil fuel combustion or industrial agriculture have exceeded the capacity of vegetation to take it up. But these metrics were developed in places like Hubbard Brook, so in temperate systems where precipitation coincides with the growing season and where plant roots are more hydrologically connected with mineralized N. So in arid systems, we know that nitrogen pulses to streams occur seasonally and after fire, even when systems are not nitrogen saturated. So a question is, how can we assess when they are? And so I'm working with this group of people here, and our goal is to improve our understanding of how nitrogen is cycled in dryland watersheds and use that information to improve the way these processes are represented in models. And if I have time, I will give a quick plug for some of my students. My PhD student, Katie, is working on examining how nitrogen cycling, export, and plant community dynamics vary along an aridity gradient following fire in the Ruby Mountains in eastern Nevada. Mary is involved in a really cool collaboration with the Forest Service where they're actually installing temperature sensors and collecting soil samples in the path of advancing wildfires. And then using that information to understand how soil heating or fire intensity influence microbial dynamics and N cycling. And then Ray is just starting a project working on improving our understanding of surface fuel decomposition and how that changes over time, which can help us understand fire risk over time and how that might respond to things like fuel treatments. And so I guess with that, I'm happy to take any questions if there's time.
So if anybody has any questions for Erin, go ahead and enter them in the chat box. I do have to jump to Jim's presentation next, just so we can give him the full amount of time. Any questions that we do have for you, Erin, I will ask during the panel discussion. But thank you so much for presenting with us today. We really enjoyed that. Also, your cats are really cute, so shout out to the cats. So Jim Archuleta is our final speaker for the conference today. He started his career with the Forest Service in 1995. He has worked as a district soil scientist in the Western Oregon forests and as a forest soil scientist in the Eastern Oregon forests. His experience with soil survey mapping contracts and 2 patents for soil restoration equipment helped Archuleta win the Forest Service 2011 Field Soil Scientist Award. Since 2018, he has been the Regional Biomass and Wood Innovation Coordinator for Oregon and Washington. Today, he will be discussing organic matter in forest restoration and soil health. So James, over to you.
All right, great. Let's see if I can get the right presentation up and going, and we'll take a shot at it. And I'd also like to thank everybody for giving me a little bit more time and consideration in getting this set up and tying in with you guys. It's been this is a good opportunity, and actually it seems like I'll be able to take the 30,000 foot level associated with the previous discussion. So we'll see if this works out. Let me see. So now I gotta try and make show grid. Now I'm having trouble with this.
Are you wanting to make it full screen?
Yeah.
So if you go down to the bottom right, there's the little symbol that's to the left of zooming in or out. It looks like it has a stand with a little thing on top. If you click that, that should put it in PowerPoint mode.
Oh, I see. I didn't realize. Okay. No, I didn't realize it was actually working. I thought I was still trying to get it up and going. All right. There we go. So as it was pointed out earlier, I used to do some soils work, and now somebody mentioned this. Another soil scientist I know that mentioned that they're currently in recovery from soils. So I'm also currently in recovery, and often and very fortunately, I have a lot of slips in that recovery, and this is one of those. So let's kind of get into that, and I'll discuss how my current work and this soils interact. This soils discussion will interact. So just to give you an idea of how I'm going to go about this. So I'm going to talk mostly about the different types of activities that we do in the forest that may alter or detrimentally create the conditions where we need restoration. So some of those activities are harvest, whether it be the temporary or permanent access to a timber sale area. They can cause detrimental impacts. Our use of hydropower and the construction camps that are associated with those. On my first duty station at Diamond Lake, I was there in the shadow of PP&L's construction camps that were used to assemble or create a series of dams on the North Umpqua. And although the camps are no longer there, there's no buildings, there's nothing else left, but there is a compacted footprint on pumice soil that has actually done some stand alteration where we've gone from mixed conifer directly to homogeneous lodgepole. So those are detrimental effects that we want to recognize. Also, there's hard rock mining and other things that can create problems by either losing the fine soil fraction or leaving rock on the surface. Then we have recreation, whether it's developed or dispersed, all the way from rainbow family gatherings where folks get out, or hundreds or thousands of people show up in the middle of the woods somewhere and have an event for a short period of time, create enough compaction and enough soil disturbance that those effects are actually seen after the fact, even though there's some efforts to do some minor mitigations, but it doesn't always take as well. And then we have the hunting camps that are set up continually where somebody comes back season after season after season, or in some cases, folks, let somebody else know and then they take over the camp. And then we have easements also from highways, cell towers, power lines. It's not necessarily the tower or the cell tower or the power line itself. It's the easement and access into it that becomes the problem, or can become a problem. So I want to show you a little bit of what I've been talking about. So if we were to take these three monoliths, the monolith on the left, the natural soil profile, gives you a good example of what the soil may look like in at least in Diamond Lake, on the Umpqua National Forest, just north of Crater Lake. These are pumice soils. This particular soil type is Series 94. It can be 6 feet deep of pumice. You can see that there's largely no B horizon. It's all just this airfall and flow material of ash and pumice. It does have some productive activities or productive timbers that grows there, but mostly where the depth of overburden of pumice is fairly shallow, so that tree roots can actually access the buried soil horizons. Now, all three of these monoliths are the exact same soil series. So the compacted monolith, you can see, is detrimentally impacted. There's the ability for roots to go is in that one diagonal right through the center. And these samples were actually taken in 1998. And before I left that forest in 2011, I went back, and just to point out how things have stayed a problem, the compacted soil horizon no longer has that organic matter on the surface. It has been decomposed. It didn't burn away. It just consumed. Then the other thing that I wanted to point out is how we could potentially restore these types of soil conditions. So all three of these monoliths were taken within 30 feet of each other. It's the same soil series. Despite the dramatic differences in their appearance, their structure, and their color, they're all within 30 feet. The subsoiled profile, though, the monolith, that was actually subsoiled 6 months prior to being extracted, and all three were extracted the same day. So you can see that within 6 months after tillage, we now have translocation of organics have moved down through the system. It's starting to become the same color and hue of the natural profile. And while there isn't a lot of organic matter in the form of live organic matter in there, it has the opportunity. So here's the other problem within the forest. As pointed out by one of the previous speakers, biomass load leads to a problem, especially as it relates to wildfire risk. So the more fuel we have on the landscape, the potential for longer duration and higher intensity fires. While our default practice of timber sales does have a stand effect, largely the fuels that create the risk are greater than 100-hour or 1,000-hour fuels, which are greater than 3-inch in diameter. So the fuels at risk are the unmerchantable materials that we don't tend to do a lot with, other than pile them and burn them or leave them on the landscape for either a fire or to decompose. And this is a picture actually south of folks there in Utah. So this was outside of Flagstaff. And what we have was a timber sale harvest where the cost of the thinning around the area was about $6,000 an acre with a helicopter. The problem with this is there was no offtake. There was no mill to consume the wood. There was no biomass plant to consume the fuel or the slash. And that load there is covering about a 4-acre area. And in the circle, you can see for scale, you can see a forwarder in the picture. Ultimately, this pile was burned accidentally as a wildfire came through the area. I don't have the effects of the problem after the fact, but I'm sure if somebody were to go down there, they tie in. I just think it's a rather dramatic photo. And if we were to pile material on top of, let's not do it on top of a mountain where we could create hydrophobic effects that could then lead downslope. So, tying into this whole organic matter thing, we have a common denominator. So the Forest Service has excess organic matter, but when we think about the landscape beneath, farms are actually striving to build soil organic matter, and in many cases, some have too little. I know in Utah, there is some dryland farming where folks, they're planting cover crops and other types of practices to try and build the soil organic matter. So potentially we have the opportunity to use the material in the forest for the betterment downslope, giving us a landscape benefit rather than just the forest alone or the farm alone. So as I pointed out, you know, the farms have a problem, and most of the times it's not everywhere, but we have a lot of USDA agencies that are focused on increasing soil organic matter, and they are doing that by increasing the organic component of the soil, so the fresh material, the decomposing material, and the stable organic matter is what I'm really interested in, because it could potentially offer them a benefit. So one of the things that we've done in the forest is we actually applied biochar, which is where we take the unmerchantable material, turn it into charcoal, and apply it to the landscape. And as you can see, you know, it has its problems, so it's hydrophobic and can actually cause runoff. But that can also be a benefit, especially when we're applying on coarse soils. And we could use the physics of how this stuff moves to do a low tech and a low cost inoculation of that material into a coarse soil. This is the plot after the fact, where you saw in the snow where we applied it in that April photo. This is an August 10th photo, same year, 2010. Inside the plot, which you see to the left, you have grass species which are fairly coarse. They have a greater, heavier seed head. The plant matter is vividly green as opposed to just outside, where you can see that the same plant essentially is cured down to the root collar and ready to burn. So as I pointed out on the seed head for the other one, inside the plot, we have a significantly smaller seed head. As you can see in this as it's pointed out here, it's significantly larger, giving us more viable forage, potentially for big game, range, other opportunities. So that's what we're trying to do, is make this material, the unmerchantable, merchantable in some way, and land applications could be the way to go. So here are the implications. If we have the ability, and this is cannibalized from the NRCS, so we can increase the soil organic matter. For some soils, that's about 12 tons to the acre. We could potentially increase the soil water holding capacity by about 25,000 gallons per acre. For the forest, that gives us timber, range, wildlife, and fire benefits. By increasing the soil moisture, we potentially could increase tree outputs. If we increase the soil moisture, we increase the forage and animal health into the winter. If we increase the soil moisture, we increase live fuel moisture, which is a fire benefit. And then for farm management, essentially they have a lot of the same benefits, but when they increase soil moisture, they decrease crop risk failure. So once again, this is how we're trying to market this whole thing. And hopefully we're gaining some traction. And this is actually how we're looking to accomplish that. So the Forest Service has been working with Air Burners, who's a company out of Florida that had already had some existing equipment, and through a Forest Service redesign and patent application, we actually have this equipment now. Today, it's moving from Bandon, Oregon to Pendleton, Oregon today, where we're going to be doing another demonstration. We actually used this yesterday to consume a weedy, woody, noxious weed. And we were essentially turning it into a way to take the sow's ear and make some level of a silk purse for land management application. The place where we did that is the Bandon Golf Course. They're interested in utilizing that material for amending their soil, their divots, their divot mix for replacing the places where, you know, folks screw up the golf course as they're testing out their swing. And that's hopefully how we're going to be able to find these different opportunities to do that. So what we're looking to do is essentially take the land management and biomass equation within the USDA, and I speak of that because the Forest Service and the NRCS are in the same agency. So if we were to be able to have NRCS clients who are deficient in soil organic matter in some environments, couple that with the Forest Service and our surplus organic matter in some environments, we could potentially have a one USDA solution, which ultimately is just a redistribution of organic matter across the landscape. The Forest Service doesn't need to spend a lot of time burning material, which can be a high cost. On the west side of the Cascades, it can cost anywhere from $1,000 an acre or up to do some of these fuels reduction treatments. So by turning this into a merchantable product, we could reduce that cost and ultimately reduce the risk on more acres for the same dollars. And as Sonny Perdue likes to talk, our Secretary of Ag, what we're looking to do is do right and feed everybody. And we can do that potentially with the excess biomass from the forest. Willing to open it for questions.
All right, Jim, thank you so much for that. Eric McCauley would like to know if anyone has used this machine on tamarisk.
I would have to default to Darren. I know he's used it on, I think, locust and some other, no, Russian olive.
Yeah.
So yeah, and you know, once you get down to the carbon's carbon. There may be some a few elemental things that are different, or there may be some additional metals, one way or the other, but largely what we're producing is a material that's greater than 80% pure carbon.
Thank you for that. We're going to get into the panel Q&A now, but first I'm going to launch this poll. If everybody could just take a moment and answer these questions for us, that would be greatly appreciated. I'll end it in just a couple of seconds, so you can also take this time to get in any additional questions you might have for our panelists. The Q&A is really pushed by the audience, so if we want to keep this going and learn some more, we definitely need some questions. And the poll should be up there. I'll give everybody a little bit more time.
Gabrielle, this is Darren. I could address Jim's reference quickly while people are filling out the poll. And we have used a crude version of what Jim was showing. I call it big box biochar kilns. Those are available on the Utah Biomass Resources website. And it's just a simple box that we do the same method, and we have done quite a bit of tamarisk here in Utah on the Colorado River, and plan to do some more here in the next few months, as well as Russian olive and lots of other feedstocks. So hope that answers your question.
Okay, so not Shelly, Nichole, I have a question here for you. David Kelly wanted to know, would you comment on the effects, positive or negative, of light to moderate physical disturbance of surface soil crusts? Is there such a thing as senescent soil biocrust that needs disturbance to rejuvenate?
That's a really interesting question. And you know, since biocrusts seem to be so slow to develop and long-lived, we don't really have an understanding that there are any senescent communities. So as many of you who live in the West and work in the West, you'll see these late successional kind of black microtopographic crusts. But what we do know is that when you see, you know, a bike path go through or a hoof print, or that can actually, if you want to call that light disturbance, then that doesn't necessarily have as much of an impact than if you have just really gone through, as you can imagine, that if you've really gone through with a lot of disturbance. And sometimes what can actually happen is that those prints can gather water and they'll recover just fine. Or if that disturbance happens when the crusts are wet, they're much more resilient to that disturbance. But I hope this answers your question. There's not really that a senescent biocrust community that would need to be disturbed, because if these late successional crusts, once you disturb them, they can take an incredibly long time to then recover to that state.
Thank you, Nichole. Erin, this one is for you. Is anyone looking at how the impacts of wildfire on nitrogen cycling might differ from impacts of lower intensity prescribed fire or other sorts of fuel treatments?
That's a great question. Yeah, I think a lot of people are trying to kind of compare prescribed and wildfire. I mean, some of the interesting work that I've seen, and some of this is older, is trying to understand how prescribed fire can mitigate the effects of wildfire on nitrogen export. So like if you have a prescribed fire and then that is followed later by a wildfire, might you not have as much an export to streams? And so I've seen work on that by Dale Johnson in the Tahoe Basin. I think I've seen some work on that by Tom DeLuca. And this is more in forested or grassland systems. So in chaparral, prescribed fire is not typically used, with some exceptions. And that's because you have these really contiguous, fast-growing fuels. And so it's often not a practical management approach unless you're maybe trying to manage fire risk in a specific location, like around structures or something like that. Then you might have some slash burning or something. But it can be really challenging to compare the two, because in addition to just differences in heating, there's other differences with prescribed fire. So it often happens during sort of the off season, the off fire season. And in some cases, so if that is not well planned, and if it happens, like, for example, when plants are flowering or after they've flowered, that can actually delay recovery. And so you can have these other sort of interacting processes. I think I mentioned this at the end of my talk. My student, Mary, is working on directly measuring heating that happens with different types of prescribed fire. So like broadcast burns versus slash burns, and how that heating or intensity directly translates into effects on soil microbial communities. And it's a great question and has lots of components to it, but it is very hard to specifically attribute something to heating alone.
Thank you, Erin. The next question was for Nichole, but I saw that Jim answered it in the comment box. So Jim, I'm just wondering if you could address this for us right now. Dennis Will asked, can the shade be provided by shallow depth wood chip biomass?
Oh, yeah. So one of the things that we have done with the biochar is we actually combined it with at a place where we had a dredge mine. So essentially the dredge mine had removed all the fine soil fraction from the area, and all we were left on the landscape at this nearby stream were large cobbles. And when the restoration work was done to rework the surface, they actually had to perch the stream with bentonite blankets. So the stream, just to give you an idea how degraded the site was, and that caused, when they tried to plant the area with trees to shade the stream, all the plantings failed because there was no soil. So then we brought in a combination of wood chips, biochar, and biosolids to recreate the O horizon, the OI, OE, and OA. It recreated the O horizon to the point where we were allowed to support grasses, forbs, and herbs, which in turn will build the soil at some point in the future to allow tree growth that ultimately will allow shading for this stream.
Thank you, Jim, for taking the time to answer that question. Nichole, this one is for you. You answered it in the chat, but we're going to address it here too. What is the mechanism behind the improved results of shading? Could it be achieved with mulch?
That's a really good question. And so some groups have been looking at different ways to shade, because what we did in those experiments is that we took shade cloth and put it on poles above the soil, which is not the easiest thing to do. But some groups, and I don't have those data, the results in front of me, or if they are, it's coming out of tried like very thin mulch layers, or also jute. And so what you can do with jute is laying that down and it's like a jute netting that you would lay down, and that you inoculate into that both stabilizes soils like the straw checkerboard, but also provides some shading. And so we've been thinking a lot about, so what the shading does is that it decreases the UV input and the UV radiation for cyanobacteria that don't have sunscreens to protect them. And also it alters the hydrologic cycle. It just keeps the soil moisture for longer, which mulch could do the same thing.
Thank you for answering that. Jim, this one is for you. Have you used managed grazing or regenerative agriculture to enhance the development of soils in the cobble site?
Well, and that's, I guess the long-term answer is yes. That's what we were trying to do, is reestablish the condition where the soil can be built up by the grasses, forbs, and herbs. And ultimately, that's actually the cheapest way to go about this. This was a really remote site. It's over 100 miles from any place where we could gather up topsoil or any materials. So there's not a cost-effective way to go about it, and you have to use the materials that are there to try and figure out how to restore and build the soil to a point where you can ultimately achieve the goals that you're looking for either stream shading or what other vegetative management you might have put in mind.
All right, thank you. Nichole, this comes from Mark Brunson. We tend to put our experiments in ideal settings. Do we know anything about how BSC restoration is affected by slope or aspect of disturbed sites?
Yes, we do put in ideal settings, because you can imagine we didn't want to put our inoculum on a slope that then just gets washed to the nearest local stream. So that is a really good question. And what I do know is that we would likely, if we were to inoculate with biocrusts, we would need to stabilize those soils with something. Straw checkerboards, some sort of obstructive capacity if we were trying to restore slopes, but we don't know a lot about that because we haven't really tried that risky experiment.
Thank you. And then Eric wanted to know where the straw checkerboards commercially available?
That's great. So they were developed in China, and China has a huge amount of cheap labor. And so they're not commercially available, but what you do is you just buy straw, and then you take a shovel, you lay the straw on the ground, you take a shovel midpoint in the straw and just push it into the ground. And it's like you're making snow fences, like 1 meter squared snow fences. So it's all the cost is in the labor.
Thank you. Does anybody else have any other questions for any of our panelists? If you do, oh, here's one. Nichole, does sodium absorption ratio affect biocrust development?
Oh, that's a good, I have no idea. Sodium absorption. I'm not sure.
Fair enough.
That's one of those where I can look it up.
If anybody else has any other questions, feel free to submit them now. Otherwise, I'm going to hand it over to Darren for our closing thoughts for the day.
Thank you, Gabrielle, and thank you to all our attendees and panelists for all those great questions and all that great discussion. We really appreciate it. It's been a great session. Appreciate everybody's participation today. We've gone from soils in the desert to mountaintops to the forest and even golf courses. Thank you, panelists, for covering all the different kinds of soils and different ways that we can restore them. It's really interesting learning about biocrusts and how you can regrow them. Thank you, Nichole. And I was really interested, maybe not everybody saw a little discussion question I had with Nichole. Under ideal conditions, she points out she can reclaim and restore some of these soils in as quick as 1 to 2 years. I was really surprised by that. It's pretty encouraging, I think. Great to have Erin's point of views and learning about fire and how it can move nitrogen down slope. I've been on a lot of fires and had no idea about any of that. It's pretty cool stuff. And of course, Jim's input is always valued, and how we can use biomass and use some of the material from the mountains and bring it out onto agricultural lands. All great ways of restoring the soil. Thank you very much. We want to give a virtual thank you and clap to our panelists for today. I want to encourage everybody to please come back tomorrow when we're going to continue our discussions, be focused on aspen management tomorrow. And we have some excellent speakers once again. We welcome back Doug Shinneman to our stage, so to speak, this time our camera, from the U.S. Geological Survey in Boise, and he's going to talk about the role of climate and fire in shaping aspen. Really excited to have Mark Creeder, a student, graduate student here at USU, who's been learning all about aspen and how it responds to fire and its natural regeneration abilities. Some real surprises in that presentation, I think, for all of us tomorrow. And Shelley Deach from South Dakota Department of Game and Fish talking about Black Hills aspen treatments. Thank you very much, Gabrielle, and to our committee for all the work for today. And once again, to today's panelists. We appreciate everybody's participation in Restoring the West Conference. We hope to be back live next year. But in the meantime, we'll look forward to seeing you again tomorrow at noon. Thank you very much.
Thanks.